big picture concepts about cells. Okay, big picture concepts. And then we'll talk just briefly about the nucleus. And that's all we're going to get to today. So we're only going to do about 15 minutes here real quick. We'll finish up chapter 4 next week. So again, that mastering biology assignment that's posted, you really can only do the first few questions if you want to. It's there if you have a long weekend and maybe you'll be bored. Who knows? So, overview. All organisms are made of cells. We call that the cell theory. Okay, cell theory. Why do we call it a theory instead of a fact? Well, I didn't go and look at every single living thing on the planet in order to check that indeed it's made of cells. And there's new living things born all the time. I haven't checked those either. So it's a theory. Okay, that's why we call it that. Cells are the simplest collection of matter that can be alive. And we can have both eukaryotic and prokaryotic single-celled organisms. Okay, that's kind of weird for people to think about because oftentimes when people think about eukaryotes, they think plants and animals. Like, how can you have a single-celled thing that's eukaryote? Well, like yeast. Yeast is awesome. It helps us make things like bread and beer, right? So eukaryotes are single-celled as far as yeast is concerned. Prokaryotes, all of them are single-celled. So of course eukaryotes can be multicellular or unicellular. Okay. The prokaryotes, bacteria, and then something called archaeans you may have heard of. Okay, archaeans are just kind of bizarre bacteria that live in strange places. We'll talk about them a lot later. But they like deep sea vents. They live in Yellowstone, that kind of place. They smell bad often, which is impressive for a unicellular organism to produce a smell, but they do. Multicellular organisms are composites of specialized cells that could not live long on their own. So this is important to kind of keep in mind. So I am made up of literally millions of cells. Okay, literally millions of cells. And my millions of cells all have the exact same DNA in them. Whether my cell be a nerve cell in my brain or a toenail cell in my toe, they have the exact same DNA. But they're very specialized cells. They do very different things. And those specialized cells cannot live long outside of the rest of me. Okay? And that kind of makes sense, right? Because the nerve cell by itself on the table is, maybe it can dance a jig, but it's not going to last long. Okay? So kind of keep that in mind. What you are is a composite of lots of specialized cells that couldn't exist for long on your own. The cell is the basic unit of structure and function, and they can differ substantially from one another, but share some common features. So we skipped 4.1. 4.1 is about microscopy, if you're wondering. So we launched right into 4.2 in your textbook. Eukaryotic cells have internal membranes that compartmentalize their functions. So again, we're going to compare these prokaryotes and these eukaryotes. And a lot of times, when we talk about what makes a eukaryote different from a prokaryote, the first thing that people say is there's no nucleus. That's true, but it's incomplete. Okay, true but incomplete. What prokaryotes lack is the endomembrane system. All membrane-bound organelles are absent from prokaryotes. And so that's going to be the nucleus, for sure. But that endomembrane system is also the Golgi apparatus and the endoplasmic reticulum. Okay, so there's these other components that are missing as well. So what are those other components doing in a cell? Well, those other components are compartmentalizing. And in that way, the cell can specialize within the cell. Okay? Over here, I'm going to make proteins. Over here, I'm going to modify proteins. Over here, I'm going to digest waste. Okay? Within the cell, those vesicles can be unique to a particular function. Okay? And the membrane is what allows that to happen, those regions of the cell to specialize. So prokaryotes, again, are bacteria and archaeans. Eukaryotes, more than just plants and animals, fungus, and then protists are also in that group. All cells have certain basic features. So this is everybody I've listed up here. Everybody has a plasma membrane that surrounds it. That selective barrier. Also has cytosol. You may have heard cytoplasm. We'll typically use them interchangeably. The cytosol is just the solution. The cytoplasm is the solution and all the stuff that's dissolved in it. Okay? Don't worry about the difference. They all contain chromosomes as DNA, and they all have ribosomes, which are these complexes that make up proteins. But there's still a lot of differences, and you guys read about them for that first night's quad. The location of the DNA is different, nucleus versus the nucleoid region. The contents of the cytoplasm. Really, it's the difference between having those organelles and not. And then size was another big one. Okay, the eukaryotes being much larger than the prokaryotes. why cells are so small. In brief, okay, in brief, the activity of a cell is happening on the inside. Okay, so if I want you to think about a cell like a ball, okay, the activities that you guys saw in the video are all happening on the inside of that cell. But most of these activities involve chemical reactions. We learned about that last unit. Okay, so a ton of chemical reactions. What do we know about chemical reactions? Well, they need reactants and they produce products. Okay? The products are typically waste. For example, CO2. Right? You need to get rid of that. So if all of that use of reactants and waste of products is happening on the inside of my cell, the only way for me to get more reactants or to get rid of more products is to send it through that membrane. Because that's the part that communicates with the outside world. So we have this issue. It's a mathematical one. The issue is, as a cell gets bigger and bigger and bigger and bigger and bigger, surface area gets bigger slower than volume. Okay? Let's look at why. On your formula sheet, and you don't need to flip to it, they give you some surface area and volume equations. Let's look at... We're going to look at surface area of a sphere. Just for funsies. So the volume of a sphere is 4/3 pi r cubed. The surface area of a sphere is 4 pi r squared. It looks like that. Well, let's ignore this for right now, because as the radius increases, the surface area is only increasing by a power of 2, but the volume is actually increasing by a power of 3, right? Our formula tells us this, which means as we get bigger, we have a lot more volume more quickly than we have surface area. Why does this matter? It matters because the surface area is the thing that gets the stuff we need and gets rid of the stuff we don't need. Okay? So as we think about this, we have to keep cells small so that we have sufficient surface area to support the volume. Okay? That's really what it comes down to. So now let's look at units. Again, these are just numbers, so who cares? But if I'm measuring, we're going to have a ridiculously sized cell. Oh, awesome. Just toss that in there. Perfect. Oh, thank you. So if you are measuring your cell in centimeters, this is awesome, right? And you figured out the volume of it, you would have the volume has the units of centimeters cubed. Correct? That's the volume unit. If I were figuring out the surface area, again measuring in centimeters, and found the surface area, my units for surface area would be centimeters squared. Well, in science we like to talk about surface area to volume ratio. Well, this one is the one we said was surface area, and this is the one we said was volume. So this is misconception alert. Let's not do this. Does that reduce to centimeters? That was a trick question. No. Does it expand to that?
Yeah.
Yeah, it expands to that, right? And we can cancel. This reduces to that, right? Right? Do you guys agree with me that that's true? This is just algebra. Okay, and then that can be rewritten as centimeters to the negative 1. Okay, 1 over centimeters equals centimeters to the negative 1. Yeah? Okay, here's why I tell you that. I once had a teacher call me and be like, you gave your 5 kid something that has a negative exponent as a unit? And I was like, yeah. They're like, that can't be right. And I'm like, well, it is. So, it is. Don't be fooled by centimeters to the negative 1. All that is 1 over centimeters. This is the unit. It's not going to be in centimeters. It's going to be in micrometers. It's cells, right? This is the unit that we use to talk about surface area to volume ratio. Okay, it'll be like 1.2 centimeters to the negative 1. Keep that in mind. Okay, don't be tripped up by the units. They're not really that hard when you think of it this way. Okay, but if I were to just tell you, oh, the appropriate unit for centimeters or for surface area to volume ratio is a negative exponent, you'd have been like, Excuse me?
So like volume's like 100 times smaller, like a lot smaller.
So, other way around. So volume increases much more quickly than surface area does.
Yeah, okay.
Yeah. That's what we're looking for. Okay. So this is calculated as surface area to volume ratio. Okay. That explains this surface area to volume ratio issue. We need to have a really high surface area to volume ratio. That tells us why cells are usually so small. This explains also why they often have very bizarre shapes. For example, this is what it looks like if we were to zoom in your intestine. Okay? These totally bizarre shapes, we call them microvilli. Why do they exist? They exist because what we're trying to do is for a cell of a particular volume, get as much surface area as possible to import and export the materials that we need, and that's which are waste. Okay, because we have to have those things. We have to be able to communicate with the outside world molecularly, and the way we do that is through the membrane. So we have these really bizarre shapes in order to allow that to happen. This is, like I said, your intestine. For the most part, larger organisms do not have larger cells. We just have more of them. There is one notable exception to that, and that is nerve cells in large animals like giraffes. Those actually do just get bigger. And it's for a long list of reasons we'll get into in Unit 7. But for the most part,
A long way from now.
I know that's March, but we'll get there. Okay. But for the most part, a bigger organism does not have bigger cells. Me and Penelope, the grading assistant cat, we have the same size cells. I just have way more of them than she does. Okay. So now we're going to focus on just eukaryotes. The eukaryotic cell has many membranes because we have that plasma membrane on the outside, but then we also have all these membrane-bound organelles, and that's going to be the nucleus, and the endoplasmic reticulum, and the Golgi apparatus, and all those vesicles, all these membrane-bound things. Okay. Those internal membranes are extensive and very elaborately arranged. And those membranes are what divides the cell into compartments. And those compartments is what you've called organelles. Okay? Before you've just been told to memorize them. That general membrane structure is always going to be that phospholipid bilayer. And remember, we talked about that phospholipid bilayer. What we said was there were no bonds holding this together. It was all because this is nonpolar, hydrophobic, and these are polar, hydrophilic. So because it wants to be chemically stable, it's just going to stick together. Because remember, these guys think the rest of the world is evil. And so then each of our membranes can have proteins embedded in it. For that to work, this area of the protein is going to be hydrophilic. This area of the protein is going to be hydrophilic. But we're going to have a whole bunch of hydrophobic amino acids on this area where the phospholipid bilayer exists. Okay? That's going to keep that protein in place. Those hydrophobic interactions are going to keep it along. So every type of membrane has a unique combination of these lipids and proteins, and that's what allows the membrane to specialize for its function. Okay, these proteins that we've got right here. So I promise that we're going to do just a little bit of our endomembrane system. Nucleus contains the genes in the eukaryotic cell. You probably know that. The nucleus is contained by a membrane. In fact, it's a double membrane. It's called the nuclear envelope. It encloses the nucleus and separating its contents from the cytoplasm. Okay. So when I say it's a double membrane, there's actually 4 phospholipids. Membrane, and then another one. Okay. It is perforated by pores. We saw that in the video, the little holes. Okay. And the little holes will let RNA out, either ribosomal RNA or messenger RNA, depending. Okay. Allows stuff to leave the nucleus. The nuclear side of the envelope, so the inside part, is lined by something we call the nuclear lamina. It's kind of like tent poles. If you've ever been camping and you have like the telescoping poles where you put them all out and the tent's flat and you stick them in the ends and the tent goes boom, right? You stick them in the ground, it goes. That's pretty much what the nuclear lamina is. It maintains the shape of a nucleus like by physically mechanically supporting it. So the DNA is organized into discrete units called chromosomes. And a chromosome is one long DNA molecule that's associated associated with proteins. So that's what this is here. The DNA is the little bitty blue thing. And then all of this other purple stuff, that's protein that's associated with it to package it up. The DNA molecule itself is so unwieldy that it has to be packaged so they don't get tangled with brain. So when they're not dividing, when the nucleus is not dividing, and that's mitosis, we have just a diffuse mass of chromatin. When it's dividing, that's when we condense into those chromosome structures that look like X's. You've probably seen pictures of those before. So in that non-dividing nucleus, there's also something called a nucleolus. That's not a typo. Nucleolus. That's where ribosomal RNA is synthesized. Ribosomal RNA is the stuff that leaves the nucleus, joins up with some protein, and becomes a ribosome. Okay? And then those ribosomes are going to engage in protein synthesis. So in sum, the nucleus is really the thing that contains both the directions in the form of chromosomes, as well as producing the ribosomal RNA that's going to actually engage in protein synthesis, all coming out of the nucleus. So this is the information I wanted you to have today before you actually go through the DNA extraction process. We had generally good success first period. If we didn't have success because the water wasn't hot enough. We need steam. Okay, we're looking for steam. Here's everything we have over here. And over here, don't take anything away from this station. Please work with like 2 or 3 people. I have 7 lab stations set up, and your graduated cylinders are in those lab stations. So if everybody works with just one person, I don't have enough stuff. Go.
Yeah, I'll give you. Yeah.
Okay. Just do it, right? Because you should have watched the video.
Yeah.
So.
I just wanted to like a paper.
Oh, well, there's directions over there. You'll see them. Yeah, because you probably didn't memorize them. So first item of business, send somebody to turn on hot water.
Your hot water. She's pretty straight today.